So I wanted to tell you about a very recent study in the lab, and I'll get to why this links into our past Human Frontiers funding at the end of the talk. The story I wanted to tell you about rests on the work of two PhD students in the lab, Natalia Rosas, who finished in 2022, and Sally Byers, who finished in 2024. So the starting point for all of this is Natalia's project, which was to address what is considered by the WHO to be perhaps the worst superbug in the world. At this time of superbug crises, that's saying something. And this is carbapenem-resistant Klebsiella. So this is an almost untreatable infection that people get. Many parts of the world are really badly affected. Australia is starting to become affected by this bacterium. And Natalia's project was to have a look at the mechanisms of carbapenem resistance and how they evolve. There's a famous mechanism that we were not interested in, but there are a couple of non-famous mechanisms that we wanted to get a handle on. None of this matters for the talk. It's simply to tell you that since she was going to do her work with experimental evolution, the starting point was to have a really high quality genome sequence of her strain. And so she went to the trouble of doing very deep short read sequencing as well as long run sequencing. And what is important for the talk today is that she found that the strain she was starting with had a chromosome, it had a plasmid, and it had a telomere phage. So we knew about chromosomes and plasmids, and we knew that they were important for her project, and we didn't know about telomere phages. In fact, they're a sort of a curiosity where only a few papers have been published, and those papers have simply focused on the weird DNA replication mechanism that these phages have. They exist in a bacterium like, well, in this case, Klebsiella. The famous one was in E. coli, as a linear piece of DNA with telomere ends. So Natalia finished her project, and after she submitted her thesis, she did a couple of experiments just simply to see whether or not we could get the telomere phages out of her strain. And there's a classic trick in microbiology where you use the DNA damaging agent mitomycin C to wake up a phage. And she was able to show, together with Andrea Rocca, postdoc in the lab, that you can in fact get these telomere phages to package into phage virions, and then you can use cesium chloride gradients to purify and concentrate the phage. So there was, sitting in the lab, a really active and concentrated purified form of the phage that we call NAR688. I'll try not to use terms in the talk, but I'll slip into this every now and then. Important point for the talk then was this was the starting point for the next PhD student who started in the lab. And I managed to convince Sally Byers that she should work on something that had not been worked on since the 1980s, but for different reasons. So we didn't really care about the DNA replication mechanism of the telomere phages. That was all really well documented by some beautiful work that had been done previously. What we wanted to know was, could we learn something about the biology of these telomere phages? Natalia's sequencing had given us an indication, it's not perfect, but an indication that these phage exist in about 30 copies per cell. So it's a lot of DNA that these cells are carrying around, and we figured there must be some selective advantage to that. So Sally took the purified phages, she infected naive Klebsiella, and that gave her a Klebsiella system, isogenic strains that were plus or minus the telomere phages. And she then did proteomics experiments to work out what impact the telomere phage had on the proteome of those Klebsiella strains. They grew fine, but we wanted to know what they were doing to the proteome of the cells in case there was some selective advantage. And I can report that it was hugely disappointing to her that they neither suppress nor activate the expression of any of the Klebsiella proteins. And in fact, there are simply 7 of the telomere phage proteins that are expressed during this vegetative growth of the Klebsiella with a phage on board. 5 of them were for DNA replication, so that made a whole lot of sense and it keyed in with all of the past literature, but 2 of them were not. And so those 2 proteins then became the focus of Sally's project. From sequence motifs and also from alpha-fold representations, the structures shown there are alpha-fold structural predictions, it looked like these 2 proteins would be a pair where one was a toxin and the other one is a membrane protein which was consistent from some other systems that it may be an immunity protein, so an antidote for that toxin. So Sally purified the putative toxin, which we call Tel-A, so that's for telocin A, and telocin is just microbiologist slang for a telomere phage encoding toxin. Sally purified the proteins and then did dilution series on lawns of Klebsiella and could show that these were really good at killing off the Klebsiella lawns. And likewise, if you take a lawn of Klebsiella where you've deliberately expressed on a plasmid the putative immunity protein, then you do save those cells from being intoxicated. So there was good evidence then that Tel-A is a toxin and Im-A is the immunity protein, and we wondered therefore why do telomere phages provide this to their hosts? Well, the puzzle got even bigger when Sally did comparative genomics analysis, because looking at the thousands of Klebsiella genomes that are available in NCBI, it was really clear that about 10% of them carried telomere phages, so they're not curiosities that every now and then pop up in our view, but in actual fact they were something that had been hiding in Klebsiella for a very long time and we just hadn't known to look for them. And of those, well, more than 1,000 genomes where we had telomere phages, they came in 6 flavours. So there's one set of telomere phages, relatively rare, which have no toxin antidote pair of proteins. And then the other 5 have different toxins. So there's the Tel-A type that includes the phage we started with. And then there's a Tel-B and a Tel-C and a Tel-D and a Tel-E type, where each of the toxins is structurally a bit different and therefore we imagine somehow differently active. So we didn't have to imagine too long. Sally purified all 5 of the toxins and she went through a panel of more than 50 different Klebsiella strains that we hold in the lab, and she was able to show that all 5 of these proteins are toxins, but they infect with a really different range of capabilities. Some of them are able to infect almost every Klebsiella we had, whereas others have a really restrictive range in the Klebsiella that they can kill. And the checkerboard there is deliberately put there so that you can't read, because I don't want you to bother with the names of the strains, but simply the dark colours say it was complete death, the light colours say there was a large growth inhibition, and the white part of the checkerboard is telling you that those Klebsiella were immune of their own accord to those toxins. So the final part of the activity in terms of looking at toxins was a beautiful assay system that Sally set up, and this was actually one of these great pieces of science where we submitted the paper and a reviewer said you should really do this experiment because that would really be the icing on the cake, and we kind of agreed, so we decided that we'd do the experiment. The experiment that you can see here just shows you 2 different strains of Klebsiella. There's AJ218, which has been transformed to express GFP. So it glows, and when you have a look at the plates in dim light, then those colonies that are AJ218 show up as black colonies. And there's another strain called AJ292, I thought it was. AJ292 doesn't express GFP, so those colonies are grey. And then Sally went through with 2 forms, so altogether 3 forms of AJ292, the starting naive strain. Secondly, a strain that expressed the telomere phage that doesn't have a toxin, that's the middle panel. And then lastly, the AJ292 strain that expresses the telomere phage that has toxin A. And you can see, then I'm going to lose my ability to click the slides. You can see in this last panel that in fact all the black colonies are gone. So in other words, if you're a Klebsiella strain and you're bothering to carry around a telomere phage that encodes a telocin, you can secrete that toxin and it'll give you a competitive advantage against Klebsiella that don't have the immunity protein to protect them from that toxin. So at the very end of Sally's work, and it's really, you know, as one of these unfortunate things in Australia, we have PhDs that run for too short a time, so you never really get to the end of a project. She was able to start to address the question of how does a telocin get into the cell that it's going to kill. So this idea of protein import into bacteria, you know, a bacterium that can import a protein, in this case to its great peril, is something that's been kicking around in the literature for a while. There are various proteins that do get into bacteria, but it's kind of a rare thing, and it's also something that doesn't really sit right with our concept of how bacteria work in the environment. The idea that they would want to be importing proteins rather than genetic material is always kind of weird. It's likewise true that their membranes are not built for importing large proteins. We know all of the pores that are in the outer membrane of a Klebsiella cell, and there's very few of them that you could imagine could actually allow a protein in. So we wanted to try and address this question of protein import. This experiment is complicated, and I won't go through it, but it's simply to say that using a whole lot of growth conditions with a battery of different variant forms of bacteria, Sally was able to demonstrate that there is a protein that serves as a receptor. So if telocin is going to get into a cell, it has to be expressing this protein. So for telocin A, it has to be this protein, a protein called OMPK36, and it sits on the surface of the bacterium. And the coloured parts there, the turquoise, green and pink, show you surface exposed loops. They show you 3 surface exposed loops in OMPK36 that you must have. If we change those over for loops from another protein, the receptor doesn't work anymore. You have to have all 3. And the 3 of them sit together. So in space, in this porin structure, these 3 loops sit together. And so a reasonable conclusion to that experiment is that this is a binding site. It's a binding site formed of 3 small loops. They come together, and telocin A can be recognised by that site. So at the very end of her thesis then, Sally had this cartoon, or a version of this cartoon, that just demonstrates where we want to go with this. So we believe that telocin A is recognised on the surface of a bacterium, and we postulate that there are other proteins, X, Y and Z, that would form a translocation channel and maybe help with the energetics of trying to unfold and get these toxins across the bacterial membranes, so that once they're inside, they can do what they do and kill the cell that's gone to all the trouble of importing them. So I have to get another PhD student because I want to find out what X, Y and Z are. And it'll of course be a difficult conversation, or would be a difficult conversation to say, well, there's probably 3 proteins and you should go off and try and find them. I think they're there. But this is not, and here's the link to thank the HFSP, this is not the first time I've seen a conundrum like this. And in fact, when I was a PhD student, there was a paper that kicked off my PhD, which was from Jeff Schutz's group at the University of Basel, where they'd just found the first component of the mitochondrial protein import pathway. And the cartoon that we were using on the previous slide is a homage to the figure in that paper, with these classic graphics that you used to use in Nature papers, where having identified the first protein, ISP42, Jeff's lab had decided that they were going to go out after the entirety of the protein import machinery for mitochondria, and in competition with Walter Neupert's lab in Munich, a few years along, we did go from one protein that probably does a job to an entire pathway full of components, energetics, various pathways that different proteins can use in order to get inside of mitochondria. So where we go now will be an investigation to work out the receptors. So we know the receptor for telocin A, and we now know that it's also the receptor for telocin C and E, but it's not the receptor for telocin B and D. There's another protein that must do that. We want to find the translocation pore. It has to be something with a bigger pore than the OMPK type porins, because it needs to be able to let at least an unfolded protein through. And we want to investigate whether there are various forms of energetics. It might be transmembrane potentials, it might be ATP-driven motors, depending on where they sit, and try and really come up with a good grasp of the pathway for protein import into bacteria. And so I did really want to finish with a genuine note of gratitude for the Human Frontiers Science Program, because I think, apart from the fact that I had a great time as a postdoc and got this very nice award on the occasion of the 10th anniversary of the HFSPO, it was also one of these eye-opening times. And for those of you who either have gotten or will get, have gotten or have recently finished your fellowships with the HFSP, I'm sure you had the same experience as me, and as you get older, you'll reflect back on it in the way that I do. I think it really changed the way I approach science, and it also certainly gave me the courage to start thinking about, we've found one component of what will be a huge pathway, but we should go after it because it's been done before, and I was part of that. So with that, I would thank the HFSP, also the ARC and NHMRC that fund our work now, And our partners, so we collect Klebsiella and bacteriophage from all around Melbourne. Melbourne is built on the unceded lands of the Bonorong and Wurundjeri people, and we do all of our collections in partnership with them, and we benefit from their knowledge and insight as well. Thanks very much.